Assalamualaikum warahmatullahi wabarakatuh. Good evening, everyone. My name is Fitri Selvalen. I'm from University of Bangka Belitung, Aquaculture Department. First of all, I would like to express my gratitude to the ICOGEE for approving my papers and providing me to the opportunity to present my research. Today, I'm willing to share my study with the title Unveiling the Close Relationship Between Beta Burdigala and Beta Uberis through the DNA barcoding based on C1 gene. As a matter of fact, Beta Burdigala is an icon to Bangka Island. Not everyone in Bangka knows about it, but the world knows. Beta Burdigala is an endemic species which just only exists in Bangka Island. This species has been sold in global aquarium trade for decades with a high value rare, which is a good news. Anyway, there is a piece of bad news too. Beta burdigala was listed as a critically endangered by the IUCN Red List and threatened species. The population of Beta burdigala has decreased because of thin mining effect and human activity, which destroying the natural habitat of Beta burdigala. Furthermore, the hunters also responsible for the population decline in the wild. The point is, Beta burdigala has a high value, but the population was declining. How about Beta uberis? Similar to Beta burdigala, this particular species only occurs in Borneo. However, its population also declined while being exposed to international trade. Beta burdigala and Beta uberis are the wild beta from the Osmobromidae family, beta genus, and Coquina group, which is they are really close. At the first glance, they look very similar with the red wine color, but there are several significant signatures between them that make them different. To confirm whether the species are actually the same or different species, we carry out the molecular analysis using the DNA barcoding method. DNA barcoding can be used as a tool for rapid species identification by using standardized genetic regions, one or more genes in mitochondrial DNA. Then, we use a standard gene for fishes called the cytochrome C oxidase subunit 1 gene. The purpose of the study is to find the percentage of similarity between those species, then analyze the polymorphic site, the genetic distance, and presenting the evolution using the phylogenetic tree. This investigation will determine whether Beta burdigala and Beta uberis are the same species or not. Obviously, we have a standard for each species identification analysis. I know there is a big question here, whether Beta burdigala is only found in Bangka Island and Beta uberis is endemic to Borneo. And is it possible that they are from the same species? The answer is why not? It's very possible. Let's learn some history because learning freshwater fishes is half of learning history. Sundaland is a region of southeastern Asia for million years, which cover Bali, Borneo, Java, and Sumatra and their small islands. The rivers between those islands were connected until the second maximum glacial era. That's why those islands have a similar freshwater fish. The factors suggest that Beta burdigala and Beta uberis are the same species or related to a common ancestor. And then let's talk about the method for attaining objectives. We obtain wild Beta burdigala from a pet swamp stream of Bikang Toba Ali area, South Bangka Regency, Indonesia on April 2023. To get the DNA barcode of species, we carry out a DNA extraction, a process to get the whole pure DNA. And then we did a PCR method to copy the DNA target to make the DNA target become millions. And then we do the electrophoresis to assess the quality of the DNA. And then you will do the sequencing analysis to read the target of the DNA sequence. And then the sequence of beta ovaries were obtained from the NCBA gene bank. We calculate the similarity of those species using the BLAST-N method in NCBA gene bank. It's an open access. Differences in the nucleotide basis of the species can be identified by conducting polymorphic site analyzing using the MEGA-10. The genetic distance was calculated using the neighbor joining method with the bootstrap test about 10,000 replicated. The evolutionary tree was computed using the maximum composite likelihood method in MEGA-10. Also with the replicate 10,000 bootstrap method. And this is the DNA barcoding result. We have a DNA barcoding of beta burdigala from PET swam of Bikang River, Tobo Ali district, and also the DNA barcoding of beta uberis from the Borneo. We get this from the GenBank NCBI. This is the research finding. DNA barcoding and species identification. Beta burdigara from Bangka Islands, Indonesia, have 670 base pairs, and beta uberis has a 665 base pairs. However, the minimum of the 600 of 58 base pair long fragment using the CO1 gene can be used as a basis for differentiation between animals and species identification. Well, it's standard. The sequence of beta burdigala from Bangka has around 96.66% similarity to beta uberis from Borneo, which is almost 97%. According to Hebert et al., 2003, said that similarity about the 97 to the 100% means the species are identical, and the species with differences up to 3%, 3 or 4, 5, and % is a different species. So in here we have the similarity between beta burdigala and beta uberis, where 96.66%. So which is the beta burdigala and beta uberis is a different species, actually based on the DNA barcoding CO gene. And how about the polymorphic sites of beta burdigala and beta uberis? Differences in the nucleotide base of the species can be identified by conducting polymorphic site analysis. There are active nucleotide bases different between beta burdigala and beta uberis. The mutation were dominated by the transition mutation A to G and C to T, a 17 mutation of transition mutation, and 1 transferation mutation, G to C. And how about the genetic distance between the beta burdigala and beta uberis? The beta burdigala and beta uberis have a close genetic distance than the other beta incognita group. Genetic distance is a degree of differences in a gene which is calculated based on differences between species or population. The closest distance between species occurs in beta burdigala and beta uberis about 0.04, which is mean that out of 100% of base pairs, there are 4 different base pairs between them. According to Ney, 1972, a genetic distance of 0.010 to the 0.099 is included in the low category. So the beta uberis and beta burdigala really close with the low category genetic distance. Then the others, beta in the same group, which is the beta burdigala and beta uberis, belongs to the coquina group. This is the evolutionary history. Beta burdigala and beta uberis are the same clade in phylogenetic tree. This is the beta burdigala and this is the beta uberis, and this is the coquina group. So in coquina group, beta burdigala and beta uberis make a new group. So this is a unique result. This is I think that this is the big result from the C1 gene, beta uberis and beta burdigala, separated to the coquina group and make a new group. And maybe we can call the burdigala group or the uberis group or anything. So beta burdigala and beta uberis have a common ancestor. So this is the ancestor of the beta burdigala and beta uberis. Then they are separate. Beta burdigala went to the Bangka Island and beta uberis went to the Kalimantan from the glacial era too. So they are really close in genetics. Maybe that because of the environmental differences, so make them different. This indicates that a close evolutionary history and family tree. This supports our hypothesis that these species are morphological similar, which class them together in one group. So if beta burdigala and beta uberis not in a same species, so it's really different species based on morphological and based on genetically. So beta burdigala and beta uberis really close and make a new group. So we found something new in here. However, further evidence regarding of the evolutionary history and genetic of beta uberis and beta burdigala still required. Regarding comprehensive understanding, we suggest analyzing the entire genome to determine the extent of similarity between these two species. So we have to go to with the new research with the whole genome to read really clear about the relationship between beta burdigala and beta uberis. This is the conclusion. So beta burdigala and beta uberis are closely or related according to the study based on C1 gene. The sequence of beta burdigala and beta from Bangka Island and beta uberis from Borneo share the 96.66% similarity of sequence, which is a different species but really closer. So can make a new group actually. So group in a group or a different group from the coquina group or a group in coquina group. That's really possible. The evolutionary history of beta burdigala showed that a close kinship with the beta uberis, with a genetic distance of 0.04 of 687 base pairs. Those species differs by 18 nucleotides. Transition mutation dominated. The mutation dominated by the transition mutation about 17, and transversion mutation about 1 transversion mutation. The study also notes that the ancient river Sundalands connect Borneo and Bangka Island, explaining the common biodiversity. But more research to understand the beta uberis and beta burdigala genetics and evolution is needed. For complete understanding, we recommend analyzing the whole genome to compare these two species. This is our reference. Our main reference is comparing the genetic Mystacilus marginatus and Mystacilus padangensis based on cytochrome C oxidases unit 1. It's Iranian Journal of Archaeology 2023. We would like to express our gratitude to the research and community service institution of Bangka Belitung University, and also PT Timah Tbk Indonesia for funding our research and publication. We also thank to the international conference, Green Energy and Environment on 2023, ICOGEE, Universitas Bangka Belitung, Yayasan Ikan Endemik Bangka Belitung or the Tanggokas, and also Universitas Airlangga and Universitas Indonesia for the great collaboration. So this is the final presentation of me. If you have a question, you can comment in below so I can answer really fast response. Thank you for the watching this video. This is a simulation of international presentation of my research. So I'm going to put this